Today we will be summarizing Chapter 7, Origins of Species, a summary by Berena Hernandez, Daniel Hicks, and Carolina Rico. This chapter is from the book Why Evolution is True by Jerry Coyne. Ernest Mayr was a German zoologist who had a love for birds and tremendous enthusiasm for his field of study. After graduate school, he realized he lacked the field experience. Lord Walter Rothschild financially supported Mayr for an expedition to Dutch New Guinea. Rothschild owned the world's largest private collection of bird specimens and recruited the help of Mayr to help add more specimens to his collection. His expedition led him to Dutch New Guinea, and during his expedition he dealt with many difficulties like bad weather, untrustworthy pathways, constant sickness with lack of antibiotics as aid, and xenophobia of the locals. In his expedition he discovered many species, including 26 species of birds and 38 species of orchids. This expedition is what made him want a career as an evolutionary biologist, and he continued his career until the age of 100. One of his best findings was the evidence that discontinuity of nature is not arbitrary but objective facts. What he noticed in his expedition was that the locals of New Guinea Arfak Mountain recognized 136 species, and Western zoologists recognized 137 species. How can two completely different cultures find such similarities in species? This is because we tend to cluster groups of animals and plants to discrete groups known as species. Organisms within the group can easily be distinguished from one another. Take, for example, a lion versus a cougar. They are two species of organisms that belong to the cat family. However, if either one of these cats was presented to you, you would be able to distinguish a lion from a cougar based on their features. This idea caused a big problem with evolutionary theory. So what is species? Species is a group of individuals that resemble one another more than they resemble members of other groups. This is considered the morphological concept. It was how Linnaeus first classified species in 1735. So why is this definition so wrong? The problem is that it doesn't account for species with sexual dimorphism. This is when the male and female can look very different. For example, male peacocks have feathers that are vibrant hues of blues and greens, while the female peacocks are just brown. They would be considered different organisms based on the definition of species. The other problem is variation within the species. Take, for example, humans. We look very different eyes, different shades of skin, yet we are considered the same species. The reason for this is because humans, even though are very different in appearance, they are successful at producing fertile offsprings with one another. Another problem with this definition is that there are species that either look exactly alike or nearly alike, but are considered different species. These species are called cryptic species. Take, for example, the Drosophila. Females of different species can't be told apart even under a microscope, and the males can only be told slightly apart by the shape of their genitals. Another example would be the Anopheles gambiae. These mosquitoes are characterized based on where they live and which host they bite. Some of these mosquitoes are malaria-carrying mosquitoes, which could be deadly, while others are harmless. So why are they different species? Species are different because of their reproductive isolation. The species do not exchange genes, and they constitute distinct gene pools that do not intermingle. This reproductive isolation can be verified in the laboratory. This is why, even though humans look different, they can be classified as the same species because every human belongs in the same gene pool. Species are categorized based on their barriers that prevent them from interbreeding with another species. In 1942, with the help of Theodore Dobzhansky, a Russian geneticist, Mayr proposed the now gold standard definition of species. The definition states that a group of interbreeding natural populations that are reproductively isolated from other such groups. This definition is known as the biological species concept. Reproductive isolation means that different traits, like the appearance, behavior, or physiology of the organism, can prevent them from interbreeding.
Some of these reproductive barriers include mating or flowering seasons don't overlap. Just like corals, they spawn several hours different than other related species. Animals have different mating displays or pheromones. Like Drosophila, they have a chemical on their abdomens that males from different species do not find attractive. Another barrier is separation of different habitats. For example, the mimulus monkey flower has two species that share the Sierra Nevada. However, one of the species is pollinated by hummingbirds, while the other species is pollinated by bees. This speciation occurred through geographic isolation of their common ancestor. They are also considered sister species, which are closely related but do not reproduce with each other. If two species try to mate, for example, a horse and a mule, the fertilization might not be successful, or they can create a hybrid. The problem with this is that the offspring can come out sterile. This is thought by evolutionists to be a byproduct of an evolutionary accident. Species are a reproductive community. This means if a good mutation arises that produces an increase in offspring, then the gene may spread throughout the entire species. This gene, however, would not be dispersed out to any other species. This is how species maintain appearance and behavioral differences between other species that habitat the same area. Evolutionary divergence results from these mutations spreading through a reproductively isolated community. Biological speciation occurs between two isolated populations of a single species. Species can slowly diverge in reproductively isolated populations due to lack of interbreeding and thus genetic exchange. During biological speciation, populations tend to change genetically and so drastically that their own members cannot recognize each other as mates, or their genes can no longer make fertile offspring. The biological speciation concept cannot describe asexual speciation. This is because reproductive isolation does not apply to asexual species.
The biological species concept also does not apply to extinct animals. Since reproductive compatibility can't be tested, museum curators and paleontologists go back to using the morphological species concept to classify extinct animals. Specimens are classified by how they're similar and what their appearance might have been. The biological species concept is still the dominant one used, however, when studying species. A common precursor to the origination of species is geographic isolation. The theory of geographic isolation states that the evolution of genetic isolation between populations requires that they first be geographically isolated. By spatially splitting a single population into two genetically isolated parts, lack of inbreeding makes divergence inevitable. Without isolation, selection that could drive populations apart has to work against the interbreeding that constantly brings individuals together and mixes up their genes. A clear example of speciation is Drosophila. Drosophila species pairs are segregated by three reproductive barriers: mating selectivity, unviability of hybrid offspring, and sterile hybrids. It's been predicted that this was a slow process taking about 2.7 million years. Drosophila are also an example of cryptic species, that is, species that look nearly alike but are genetically distinct. We know from the fossil record, embryology, and other data that species diverge from a common ancestor. From this, we can make several predictions. One is that geographic isolation generally occurs first, prior to speciation. A geographic barrier needs to exist for divergence to occur. Secondly, we can predict that geographic speciation is still occurring in nature to this day. Third, we must be able to find the most recently formed species in different but nearby areas. And fourth, we can predict that reproductive isolation between populations increases slowly with time. Sympatric speciation is the process of speciation without the need for geographical isolation. This process can occur in habitat islands, areas like oceanic islands that are too small for a proper geographical barrier. An example of this process occurring is in cichlid fish in 2 lakes in Cameroon. One lake has 11 species and the other lake has 9 species. All of these species are descended from a common ancestor and differentiated without the need for a geographical barrier. In animals, this is a very rare type of speciation. Unlike in animals, sympatric speciation is very common among plants, as animals cannot self-fertilize. This is a form of speciation in action, as the species changes can be seen within a human's lifetime. Allopolyploidy occurs with the hybridization of 2 different species in the same area with different numbers or types of chromosomes, but these offspring are sterile and do not contribute to speciation. Autopolyploidy, however, results from the self-fertilization to form a hybrid species with double the chromosomes of the parent plant. This gives rise to new species, as the polyploid organisms cannot interbreed with other members of the species. There are several lines of evidence for speciation. We see lineages splitting in the fossil records. We also see closely related species separated by geographic barriers, as if they had just recently undergone speciation due to geographic isolation. We can also observe new species beginning to arise as populations evolve reproductive barriers, as in polyploid plants, which generate quick speciation. In spite of this evidence, however, many creationists still do not believe that speciation occurs.